Walk into any pharmacy today, and you will face a wall of supplements screaming numbers at you. 50 billion CFUs, or 10-strained gut health. We have been sold a simplified idea that consuming a high volume of generic good bacteria will universally repair a digestive system. However, clinical research suggests that probiotics should not be treated as a single therapeutic category. Effects observed with one organism, dose, or formulation cannot automatically be assumed to apply to another. This is similar to walking into a pharmacy with a migraine and grabbing a generic bottle labeled only medicine. You wouldn't expect a random pill to treat your specific headache just because it belongs to the same broad category. Relying on high CFU counts or multi-strain marketing without clinical context is a medical gamble. The word probiotic is too broad to function as a description of medical effectiveness. To find actual results, we have to look past the marketing and identify what actually drives clinical success. To find that success, we have to look at how microbiologists actually categorize these organisms. This diagram shows the hierarchy of biological taxonomy. We start broad with a genus, like Lactobacillus. That breaks down into a specific species, and finally terminates at the bottom in a highly specific alphanumeric strain code. Simply knowing that a product contains a Lactobacillus species is generally insufficient to determine if it will be effective. Two microorganisms belonging to the exact same species may differ in their ability to survive gastrointestinal transit. One might withstand a wash of stomach acid, while its neighbor rapidly dissolves and dies before reaching the intestine. Clinical studies focus on the complete alphanumeric strain designation. This code is the only identifier that ties a specific organism to a specific clinical outcome. Substituting one strain for another, even within the same species, does not guarantee the same biological result. We can see this precision in the data for antibiotic-associated diarrhea. The strongest evidence for prevention points directly to specific organisms. Lacticaseibacillus rhamnosus GG, or LGG, and the probiotic yeast Saccharomyces boulardii. This chart shows how diarrhea incidence rates change during antibiotic treatment. When the specific LGG strain is administered, the risk drops significantly compared to a placebo. But efficacy depends on more than just the strain. It also depends on the characteristics of the population receiving it, as seen in research on infantile colic. Here, the chart tracks daily crying durations in infants given the strain Limosacrolactobacillus reuteri, DSM 17938. In breastfed infants, there is a steep downward trend in crying and fussing. However, the evidence for this exact same strain is considerably less certain for formula-fed infants. The environment in which the strain operates determines its success. This environmental factor also applies to the other bacteria present in a supplement, which makes the research into irritable bowel syndrome particularly complex. While multi-strain supplements are common, adding more strains increases the difficulty of isolating which one is helping. A product with 20 different strains makes it impossible to identify which organism relieves bloating or pain. Because of this, researchers must evaluate single strains, like Bifidobacterium longum 35624, to demonstrate measurable improvements in abdominal pain and bowel dysfunction. A clinical benefit for one specific strain, given to one specific group, cannot be automatically assumed to work for anything else. Scientists are now investigating the gut-brain and gut-skin axes, examining how specific strains might influence conditions like migraine, depression, and psoriasis. This scatter plot compares our current levels of clinical certainty. On the left, we have tightly clustered data points for established gastrointestinal treatments. On the right, the findings for neurological and skin conditions are currently highly variable. These areas remain exploratory. When you look at a supplement label, blur out the broad marketing terms. Ignore the generic promises of gut health and the massive billion count numbers. Instead, identify the exact alphanumeric strain and the dose used. Clinicians and consumers must match the specific strain to the clinical outcome studied in trials. Ultimately, a probiotic should be judged by the evidence for that exact organism and exact condition, rather than by the general reputation of probiotics as a category.